Okay, hi everyone, how are you? It's 8 a.m., so again, thank you. I'm very grateful that you're putting the effort to, keep up with the course, come early. So clicker questions starts counting for extra credit today. No stress because everything is fine, any answer is fine. You need to make sure you answer the clicker when it's posted. Okay, we will do, as always, a little bit of a review and discussion, and I'm happy to answer any questions you have from lecture 4. How many of you have watched the video? Okay, thank you so much. As I always said, I would be doing a disservice and would be disrespectful for the people who watch the video if I go over the lecture. So that's not the purpose today. The purpose is to do a summary, answer questions, and use the time that we have here to practice and ignite some discussion, right? To practice some click with the clicker questions, okay? Just making sure you understand what is the format of a flipped classroom setting, which is very different than a standard classroom, okay? Okay, so in this video, in lecture 4, we learn about clonal selection and expansion. And so we learn that we have billions of specificity in the adaptive immune system, right? Like we know the cells of the adaptive immune system are B cells and T cells, right? They developed before seeing their antigen, that is the substance that the ligand for their BCR, B cell receptor or T cell receptor, they develop, right, in bone marrow for the B cells and thymus for the T cells. And when they develop, they leave the bone marrows or the thymus as naive lymphocytes that have billions of different BCRs and TCRs. Each lymphocyte will have one BCR and TCR that will be different from the other lymphocytes, right? But each of them will have only one type of TCR. Many copies, but then one type, okay? Okay, so and this will be naive, which means that they haven't seen the ligand for this BCR or TCR, that is the antigen, right? And they will, once they see the antigen, right, like we have infection with a virus, for example, like let's say measles virus, the sequences of amino acids in the glycoprotein of measles virus, maybe the ligand for one of the lymphocytes. Imagine you guys are the lymphocytes, the naive lymphocytes, and imagine you have this BCR or TCR, right? And you always have the same shape. And you have many arms and many hands, but they are always in the same shape, remember. Each of you always have it in the same shape. But it's different shape than your neighbor, right? And so only one of you or few of you will be able to bind to sequences of amino acids in the measles virus, right? And that want to be capitalized, right? Once we have a hit, we want to capitalize it, and we want to clone these B cells that have the right receptor that is able to bind to a part of the pathogen that has been invading. And that proliferation that occurs with a differentiation process is called clonal expansion. And so the clonal expansion will take one clone that is able to bind, will copy, and is paired with differentiation. What is differentiation? The naive lymphocyte, as it is proliferating, it always starts a transcriptional program that converts these naive lymphocytes in effector cell. And so basically this transcriptional program is the activation of many genes that will now allow or optimize the effector function of these lymphocytes. So when the clonal expansion and which is the proliferation and the differentiation has ended, now we will have not only many, many more of the lymphocytes with the right BCR or TCR, but they will be very fit to exert the effector function, which is always tailored to fight the pathogen, correct? So it makes sense. And that process is slow, and that's why the adaptive immunity is slow. Okay, any question about that? Yes, please.
So is it that there's like so I understand how the pool works and then they're selected and then they're cloned.
Exactly.
Is the initial cell that gets cloned, does it then return to the pool or does it also go into the body to do the immune function?
It goes into the body to exert the immune function. It's not naive anymore. So good question. It goes into the body to exert the immune function, but also, and we will learn this in one of the last lectures, few of these cells will differentiate in something that is called memory cells. So the majority of them will become effectors and will go to where is the pathogen to kill the pathogen or exert the effector function, right? In whatever place, depending on the cell. But very few will take an alternative differentiation pathway that is a little bit when you take your paycheck and you save a little bit for the future. And these alternative differentiation pathways are cells that are called memory precursors, that were the ones that will generate these memory cells that are very long-lasting. They will last for life. And so when we see the pathogen again, like years later, we can respond much better. And instead of having naive cells that differentiate, we have memory cells that will be clonally expanded and differentiated into effector, which are much faster, so much better at doing the job. And that's why we don't have symptoms, because we do it fast and we do it better and we don't see the symptoms of the infection, okay, or very mild symptoms. Is that clear? Okay, cool, excellent question, good thinking. Yeah, yes? Can you say again?
Are lymphoblasts considered naive lymphocytes?
No, because blast is a cell that has been enlarged because it's proliferating. And so lymphoblast is a lymphocyte that is in the process of proliferation. It's in the middle of this clonal expansion. And if it is in clonal expansion, it's because it already saw the ligand for their BCR or TCR, which is the antigen. Once they see the antigens, they are not naive lymphocyte anymore. Good question. Any other question? It started being fun, immunology, when you understand it. And I was telling one of the students in the office hours, you need to make sure it makes sense to you. Because that's when it's fun, and you will remember, because it has to make sense why this is happening, okay? And if it doesn't make sense, that's fantastic too, because you are learning. And so you need to ask, hey, this doesn't make sense to me. Why is this happening, And it might be that, we just don't know. And I'm happy to say if I don't know, I'll try to answer to the best of my knowledge, but I may not know, or it may not be known yet. And that's okay. It means that you have a wonderful question for scientists, or maybe for yourself to answer in the future. So, but yeah, so make sure that always makes sense, because that's when it's fun to study it and when you will remember it, okay? Okay. So we said there are two major types of adaptive immune cells, the B cells that produce the antibodies, and because antibodies are soluble, this has been called traditional, historically, humoral immunity. We typically don't refer to that anymore like humoral immunity, but I'm giving you the terminology just in case you read it as such. But it's also antibody-mediated immunity, because the B cell, which is the B cell effector cell? The B cell is naive and then differentiated into effector cells. And what is the effector cell for the B cell? Plasma cells. And what do they do? secrete antibodies, are an antibody factory, okay? And this that's why the B cell responses or B cell immunity is called also antibody-mediated immunity, because it's really the antibodies, the plasma cell is the effector cell that will produce the antibodies that are the ones that will exert the effector function. What is the effector function? How the antibodies fight an infection. There are 3 ways that we have learned already. Yes. 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 Excellent. And complement fixation, right? Remember that the classical way for activating complement is with an antibody bound, the complex. Antibody bound to the antigen. That's what activates the complex. The antibody alone doesn't activate the complex. It needs to be bound to the antigen. Okay. And the T cells are called the cellular immunity because the T cells do not produce antibodies and they exert their effector function by killing, for example. Or they produce cytokines that help macrophages in the case of CD4 T cells, and this is coming here. How do they recognize pathogen, these adaptive immune cells, and how do they work? The B cells recognize pathogens through the BCR, B cell receptor, and they recognize the antigen, which is the ligand of the BCR, in a native form, right? Like as the innate receptors, they recognize their ligand in their native form without degradation or anything, right? And they produce antibodies that, as I said, can neutralize, opsonize, or fix complement to destroy the pathogen or to neutralize and opsonize. And then we have the T cells that have the TCR, but the TCR does not, the T cells do not recognize antigen in their native form. The antigen for the T cells needs to always be presented in MHC molecules, okay? So imagine the MHC molecules are like the trays, right? The T cells will not eat if they don't have a tray in front of them. It's the only way. The same thing when they are effector T cells. If they go and let's say they go to a tissue that is full of pathogen, but is no pathogen in a tray, so not in the MHC molecules, they will not see the pathogen, okay? And so these trays, right, like these are MHC molecules, and they present peptides of the antigen, little peptides derived from antigen proteins, which already tells you that the T cells can only recognize proteins because are the only ones that can generate peptides, right? This is the only thing that the T cells eat, right? And who is presenting these trays, these MHC molecules? APCs are the professional antigen-presenting cells that have MHC class 1 and MHC class 2, both, because there are 2 types of trays, you know, let's say 2 colors, MHC 1 and MHC 2. The CD8 T cells only eat from the MHC 1 tray. The CD4 T cells only eat from the MHC 2 tray, and they don't eat from the other trays, okay? So they are very picky, okay? And the waiters that are presenting these trays are the antigen-presenting cells that can have, these are the professional waiters. They have both trays. They have MHC class 1 and MHC class 2 in their surfaces. So they can, you know, present antigens to CD4s or CD8 T cells. However, every cell in our body has also MHC class 1. So the guests that are there, they all have MHC 1 tray, you know, MHC 1, or maybe are the helpers, these ones. And they have MHC class 1. So every cell in our body has, every nucleated cell in our body has MHC class 1. And why do you think is that? Why make sense? Yes.
Since CD8 is intended to kill the infected cell, it makes sense for the cell itself to be able to tell the CD8 that it's infected.
Absolutely, exactly, right? We want to make sure we don't know which the pathogens will evolve and have evolved to infect many different types of our cells. So we want to make sure that the pathogen can be eliminated by CD8 T cells. And that's why it makes sense that any nucleated cells that can potentially be infected by a pathogen express MHC class 1. So now there is a problem. That is that there is a coevolution, right? And so many pathogens, especially herpes viruses, they have evolved means, and we will learn this later, to inhibit the presentation of MHC class 1. So basically they are infecting this waiter, but they don't let the waiter show the trait number 1 with the peptide, right? And so they are infected, and the T cells are around. And the T cells, if there are no trait, they will not do anything. They will not see. That's how they see. And so viruses have evolved this evasive mechanism that they downregulate MHC class 1, right? Or they somehow block MHC class 1 presentation of peptide, and we will learn how. And so what happens if a cell that is infected doesn't present MHC class 1? NK cells, yes, excellent. And so we have another mean, because it's, again, evolution, counter-evolution. And so we have evolved also another mean to kill cells that don't have MHC class 1. Remember, MHC class 1 is the inhibitory molecule for NK cells, right? And so that's fantastic because now, we are kind of making it very difficult for the virus. And so if the virus downregulates CD8, NK cells now kill the infection. And indeed, in herpes virus infections, NK cells play a super important role because we have disabled the CD8 T cells, and the NK cells become, the way in which we can kill. Same thing for tumors. There are tumors that have mutations in different pathways of MHC class 1 presentation, different steps in the pathways of MHC class 1 presentation, and they don't present MHC class 1 with the tumor antigens. And so again, in those cases, scientists, and there are therapies that are being developed to use NK cells to kill those tumors. Okay, any question? Yes, please.
So if MHC is always expressed for non-infected cells, are they just like empty on the surface?
Oh, I love it, because this is when you start asking the questions that will come, the answers will come later. I give you a preview. They are not empty. But you know what? They have endogenous peptides. They have self-peptides. And does it make sense? It makes sense? Okay. They have self-peptides. They are always, the cells are always presenting this MHC class 1 trait, right? But they have with self-peptides, because that's what they have inside. So they present what they have inside. You might be asking why CD8 T cells don't kill them, if they have, But then remember, there are different answers, and maybe I'm getting a little bit too ahead of myself, but very quickly. So the T cells that recognize self-peptides and the B cells that recognize self-peptides are, for the most part, and we will learn, eliminated during development. And there are other mechanisms that also complement that. And so we have several mechanisms that prevent self-reactive B cells and T cells to kill or generate antibodies against self-components. Okay. Yeah, I'm getting very ahead of myself, but you will learn that, and it's fascinating how it happens. Okay. So the T cells recognize peptides in MHC molecules, in APCs, and we have two types of T cells. We have learned CD4 T cells that recognize peptides presented in MHC class 2 and are called the helper CD4 T cells. Why they are called the helpers? Yes? Exactly, because they help other cells. They help macrophages enhance their antimicrobial activity, right? By releasing cytokines in the very intimate interaction with these macrophages. They will enhance their microbicidal activity. Excellent. And which other cells do they help? Yes. That's not the typical, I would say. But good try. Which other cells need T cell help? Another adaptive immune cell. Yes, exactly. Yes, excellent. So the B cells, for the most part, in the majority of the cases, they need T cell help. And so the B cells also are professional antigen presenting cells. And I think that you were right. The professional antigen presenting cells are dendritic cells, B cells, and macrophages, monocytes, which are the precursors of macrophages. And that also should make sense to you, because if I need to be helped by a CD4 T cells, and I just said that the CD4 T cells don't see me if I don't have the MHC class 2 trait, right? It makes sense that the cells that require help from the CD4 T cells to improve, to be better, or to function, express the MHC2, right? And these are B cells and macrophages, or the macrophage precursors, which are monocytes. Dendritic cells also need to express MHC class 2 for this to work, because remember that I mentioned in the previous lectures that dendritic cells were the bridge between innate and adaptive immunity, and if you don't have dendritic cells, you don't have initiation of the adaptive immunity. So the T cells, to become from naive, be selective like that, and become the clonal expansion, for that step, they need to see the antigen, correct? Going from naive to be clonally expanded and become effectors. We're on the same page, right? They are naive, they have the receptor, they need to see the antigen. But the only cells that have the capacity to give them all the information and activation for them to go into clonal expansion and differentiation are the dendritic cells. And so the only cells that can select that clone of naive T cell to become a effector is a dendritic cell. Is that clear? And so now, if dendritic cells don't express MHC class 2, which they do, but let's say that we have a mutant in which dendritic cells don't express MHC class 2, will you have any activation of CD4 T cells? Guys, yes or no? No, right? No. Will you have any activation of, or will you have any impairment or limitation in the B cell responses? Yes, because we just said that for the most part, the B cells require CD4 T cell activation and help. And if you don't have the activation of a naive CD4 T cell by a dendritic cell that expresses MHC class 2, you will not have the help for the B cells, and the antibody response will be very limited, very, very limited, okay? Makes sense? please stop me because we're integrating a lot here, okay? So stop me if you need me to rephrase or you have any question. Okay? So now, can I ask you one question? Let's see. I'm gonna see if you understood. We said the CD4s will become from activator, a naive, clonal expansion and differentiation. Here they will be effector CD4 T cells. So when is a CD4 T cells able to help the B cell? When it's a naive or when it's an effector CD4? Effector. And which type of effector CD4 is the one that helps the B cell? Follicular helper cell. We have this differentiation, especially for the CD4s, and well, a little bit for the CD8s, but especially for CD4s. We said they are clonally expanding, and at the same time, they differentiate, which means that there is a transcriptional program, activation of many genes. This differentiation, right, is like almost I have different ways to differentiate into different things. I will always clonally expand, but I may become a follicular helper cell, or maybe I took this other differentiation path, and I will become a T helper 1 cell that will be more tailored. I activated genes that are better fit to help macrophages, okay? While the follicular helper cell, this path of differentiation, activated genes that are more tailored to help B cells, okay? So now I will ask you another important question. And this is a guess, because I don't think that we have mentioned this. So which cell is telling the T cell, the CD4 T cell, which differentiation pathway that they should go? What would make sense? Yes?
Probably the dendritic cell, because that's what activates it to do it.
Yes. And remember, excellent. It's the dendritic cells here in this initial interaction. And remember, dendritic cells are the generals, right? Are giving them all the information on what they need to differentiate. And they will tell them to differentiate and go through the path, and we only did 2 now, but there are more, and we will learn more paths, to differentiate into effector cells, Th1, or T follicular helper cells, which are better fit to fight the type of pathogen that is invading, okay? And now another question. So the dendritic cell is telling the T cell, you have to do, okay, all the instruction, okay? And then how the dendritic cell know all this information? What would make sense? Yes? Excellent. They can travel. They are coming from the site of infection with that information. Exactly. So they have been where the pathogen is, right? They are coming from the food that is infected to the meeting place, which are the secondary lymphoid tissues, where they will instruct the T cells. Excellent. So now, the dendritic cells, how they know which type of pathogen is, how they sense the pathogen. Now we are in the site of infection, okay, where all the pathogen is, the dendritic cell is surrounded by pathogen, maybe even inside. How do they know which type of pathogen we have? Yes? Excellent. TLR, RIG-like receptors, all the pathogen recognition receptors that in combination, and we still don't know exactly that language, in combination, SIGAs as well, will activate certain transcriptional program in the dendritic cells that now, while they travel, all this protein and this information is being translated into these genes are being transcribed and proteins are synthesized. So when they tell this instruction of the T cells is through cytokines and through molecules that are interacting in this intimate interaction between the dendritic cells and the T cells, okay? And that not only will make the naive T cell clonally expand, but will make the naive T cell to differentiate into whatever effector cell best fit the pathogen that is invading. And so now this effector Th1, let's say if there is an intracellular bacteria or Mycobacteria leprae or Mycobacteria tuberculosis, Th1 is the best because these bacteria grow in the macrophages and they subvert the macrophages. So the macrophages really need the help from Th1 cells for eliminating or containing the bacteria, and we will learn more about that. But so it's differentiated into a Th1, and so now this is an effector cell, which means that it's ready to fight. It's not a naive. It has all the genes that are necessary to exert their effector function. And in this case, because it's a Th1, the effector function is to help macrophages. And so we'll go through the lymph and the blood to the site of infection, where it will see the macrophages that have the mycobacteria, okay, and will help the macrophages. And how we'll know which macrophage to help? There will be many macrophages. Some will have bacteria, some will not have bacteria. Yes?
The one presenting antigen MHC class 2.
Exactly. So the only way that the T cells see is through these MHC in the cells that need to have the trait, the MHC molecules that present the antigens. And for CD4s, it's MHC class 2. Okay? They will only help macrophages that have the MHC class 2 that have the specific antigen, not any antigen, the specific antigen, the mycobacteria antigen. Okay? That is the same antigen that the dendritic cell, right, days before, have taken, put it in the MHC, in the tray, and then presented and activated this clone of naive CD4 T cell to generate the expansion of this clone. So the TCR that was selected, right, doesn't change. It will always see the same MHC class 2 antigen complex. Is it too complicated? Make sense? Okay, this is kind of a core for immunology. It needs to make sense now, because then it builds and gets more complicated. But if you remember this core, you know, you don't get lost, you know. Okay. Okay, I think that the CD8 T cells see MHC class 1, and their effector function for a CD8 T cells is to kill infected cells. And it also is to kill tumor cells, okay? We said that dendritic cells are link innate and adaptive immune system. They are professional APCs and are essential to activate T cells. So if we don't have dendritic cells, we don't have activation of naive T cells. Functional outcome of effector lymphocytes, we already talked that the B cell functional effector cell is plasma cells that are an antibody factor, and the ones that will exert the effector function are the antibodies through neutralization, opsonization, complement fixation. They need, the CD4 T cells are the helper. They will help macrophages, Th1, and B cells, T follicular helper cells. And the CD8 T cells will kill infected or tumor cells. And then we talk about the interrelationship between innate and adaptive immunity in the video. The innate immunity is necessary for and influences the type of the adaptive immunity. And we just were giving an example how the dendritic cell, right, get through their innate receptors all the information on the type of the infection and will influence the differentiation path that the CD4 T cell will take. So innate immunity through the pathogen recognition receptors will get information and will influence the flavor or the type of adaptive immunity. And the adaptive immunity empowers innate immunity. So there are two ways in which the adaptive immunity works. By empowering innate immunity, and we were just saying Th1s will go to the tissue and empower, help macrophages, right? That's one way the adaptive immunity cells work. Or they may have direct mechanisms to destroy the pathogen, like the CD8 killing, okay? So which other way in which, in which adaptive immunity empower innate immunity to fight the pathogen? Yes. The complement, right? They use the complement, for enhancing complement activation to destroy the pathogen. Excellent. Where is our immune system? And so we learn we have primary lymphoid organs, that is the bone marrow where B cells develop, and the thymus where T cells develop. Remember which is the progenitor that generates B cells and T cells? Remember? Everything needs to come together. The B cells and T cells, they derive from hematopoietic stem cells, correct? Okay, and which progenitor? Let's see if you remember? No, that's okay, no problem. Yes. Common lymphoid progenitor. So common lymphoid, they will generate the B cell, B lymphocyte, and T lymphocyte. So it's common for the two. And also will generate natural killer cells. So now, I told you, so the hematopoietic stem cells are in the bone marrow, correct? They will generate the common lymphoid progenitor. And this common lymphoid progenitor will stay in the bone marrow to generate B cells, all through the different developmental stages, which we will learn. And it will go to the thymus, okay, through the blood, will exit the bone marrow and go to the thymus, where we'll start, now in the thymus will receive signals that will start another developmental pathway that generates the T cells. Both CD4 T cells and CD8 T cells are generated in the thymus, okay? Make sense? Okay. Okay, so those are the primary lymphoid organs. Are primary because are the first one. We first need to generate the cells, right, to be able to be ready. And this is all before the pathogen, before the antigen, seeing the antigen, okay, the foreign antigen, etc., or the specific antigen. Okay, and then we have secondary lymphoid organs, which are the meeting place, right? And then what are the secondary lymphoid organs? Some examples, yes. Peyer's patches, which are in the gut, okay, in the gastrointestinal tract. What else? Lymphonodes, which are everywhere, okay, are in between our lymphatics, everywhere in our body. I think that I could be wrong, but okay, I will be careful. We have hundreds. I don't remember exactly the name. I think I have a number here, but not sure. Yes, what are spleen? And we will learn a little bit more in next lecture, so we will not get too deep into that. The similarities and differences, but all have a T cell zone, a B cell zone, and are the meeting place, because we just said that the adaptive immune cells, right, like have billions of specificities, right? Again, let's go back to the example. Each of you is one adaptive immune cell that have a specific receptor, right? But if I'm a pathogen, right? Only one of you will have the or let's say in the campus, let's imagine all the students in the campus, maybe 1 or 2 will have the receptor specific that attacks me, that binds to my proteins or or to my antigens. Okay, I'm in a pathogen here. So I come to UCSD as an antigen, as a pathogen, and only 2 students in the whole campus will have the correct antigen. And I come in the library, and these students are, I don't know, in center. And so what are the chances that they will see me? By the time they find me, I'm just replicating, infecting many cells, right? And so that's why we have a meeting place, and that's Price Center. No, just kidding. And that's the secondary lymphoid tissues. And so all the students, all the lymphocytes will go and circulate, through these secondary lymphoid tissues, right? For example, in the lympho nodes, which are in all our body. So let's say this is the meeting place, this is the lympho nodes. The lymphocytes, all the students, at some point, will come through the lympho nodes, and there are many meeting places, not just one, okay? So they will always come and check in the meeting places and go walk, and then they do all this circulation. And this is basically, we'll learn more of the circulation. I don't want to get into that, but I want to get the big concept. So we'll learn the details in the next lecture. And the dendritic cells, right, will come, we just said, they will come from the side of infection with these antigens, or even pathogens sometimes, to the meeting place, so to facilitate the encounter of the T cells with their specific antigens. And also, the B cells don't need the dendritic cells, correct? Remember, the B cells don't need the dendritic cells. The T cells need the dendritic cells because they need this MHC presentation, okay? The B cells don't need the dendritic cells directly because they can see the native antigen, correct? But it's a tricky question because they indirectly do need the dendritic cells because they need the help from the CD4 T cells, and the CD4 T cells need the dendritic cells, correct? Okay. You're following me, or I'm talking nonsense? Okay. Stop me if because I'm trying to integrate. You study and I think that this is the best way to learn, to really learn. You first study very organized in the lecture, and then here we do a mess. But that's integration. And I think that that's important because that's how it makes sense, because all happens together, not in the order in which we present the slides. Okay. So the B cells don't need the dendritic cells, but the B cells see the native antigen. And so these, the dendritic cells through the lymph, right, will go to the meeting place, the lympho nodes, to present the antigens to the T cells. But the lymph also brings native antigens, okay, from the tissues all the time, not just when there is infection. All the time is the dendritic cells and the native antigens from the tissues are coming to the lympho nodes, okay? But if there is no infection, this will be self-antigens, okay? And so these native antigens that come through the lymph now will be seen by the B cell that has the right receptor in this meeting place, okay? So that's how it makes sense. That's how, 2 students or 3 students in the whole campus, if they have the right receptors, they will be able to see me because the dendritic cells or the lymph will bring me to this meeting place, and the B cells and T cells are constantly recirculating here, so facilitates that they encounter me. Okay? Any question about that? Yes?
How are foreign antigens brought from site of infection to the secondary lymphoid organs?
Excellent, excellent. How do foreign antigens, it's an exam question. How do foreign antigens are brought from the site of infection to the meeting place, which is the secondary lymphoid organs? Anyone? How are they brought from the site of infection to the secondary lymphoid organ? Yes? Good point. I think macrophages are not migratory. I mean, good try. Macrophages are not migratory. So they are staying in the tissues. They don't go to the meeting place. Or they are in the meeting place, but they don't traffic. You know, they are more statics. They stay where they are, which is tissues, any tissue. Yeah. So which other cells do traffic from the site of infection to the secondary lymphoid organs that we were just talking? What's your name? Sarah. So which cells traffic from the site of infection to the secondary lymphoid organs, to the meeting place? Which cells? Which innate cells? That are the ones that will activate the T cells. Remember that we're saying they are getting all the information here through pathogen recognition receptors, and then they will traffic to the secondary lymphoid organs to give all the instructions to the T cells, which dendritic cells. So that's one way in which the antigen in the site of infection is transported by the dendritic cells through the lymph to the secondary lymphoid tissue. So that's one way. But for the B cells, they don't have, they are not activated by the dendritic cells. So how the antigen is transported, the native antigens are transported from the site of infection to the secondary lymphoid organs? Yes? Through the lymph, exactly. You know, the lymph is that's what the lymph does. All the time brings liquid with antigens to the lymphoid tissues and recirculate and then go to the blood. And then the blood basically extravasate, and without albumin, the blood converts into lymph in the tissues. They all brings all what is in the tissue to the secondary lymphoid organs. Then it goes, the liquid goes, and again, and it goes back to the blood circulation, and then again and again and again. Yes, so through the lymph is, so antigens are transported from the site of infection to the secondary lymphoid organs in two ways: through the lymph and via dendritic cells. Yes.
Is there a specific mechanism that the lymph uses to bind the antigen before bringing it?
No, because the lymph is liquid. It's like, I'm trying to find an analogy. What would be a good analogy for the lymph? I guess that when you clean, right? When you clean something, and then you will carry all, let's say that you are trying to clean the sidewalk, and then you are cleaning, and there is a lot of liquid, and then you move all this liquid into the secondary lymphoid organs, and the liquid will have all the particles, or the dirt, or everything what was there in the sidewalk.
Okay, so it just sort of gets the pathogen just gets caught in the flow.
Exactly, yes. What else? Anything else? Yes. Yes.
So say once a B cell binds to a pathogen with the BCR, it still needs T cell activation to start producing antibodies.
Excellent.
But the antibody binds to a completely different scene of the pathogen.
Yes.
MHC.
That the TCR.
So how do you make sure that MHC, the B cell name, is the same MHC, like not MHC, but the antigen on MHC?
Oh, that you are 5 lectures advanced. Is fascinating. I mean, we don't have all the elements to ask, to answer that question at this point. It's an excellent question. So, but basically, the B cell has the VCR that recognize one part. Let's say that this is the pathogen. This is one antigen in the pathogen, right? That, let's say, that's recognized by the VCR. But the B cell will uptake all the pathogen, and the pathogen has another antigen, let's make it like that, that is recognized by the T cell. So the B cell will uptake the pathogen, the whole pathogen, right? And it will uptake it because it's able to see this antigen, right? And so this antigen will bind here, and the B cell will uptake the whole pathogen and then present in the MHC molecules the T cell antigen. And so now the T cells, through the TCR, will be able to see this antigen to help the B cell. It makes sense or not? Or is too complicated? We will learn this a lot. But in a nutshell, so this is the pathogen. It has multiple antigens. Some will be recognized by the VCR. Once the VCR recognizes the antigen in the pathogen, it will uptake the whole pathogen. So now you will have here the whole pathogen with the B cell antigen that was bound there, but also a T cell antigen. And these T cell antigens go and is presented in the MHC molecules. And so MHC2, well, MHC1 as well, but MHC2, and then the TCR can recognize the B cell that has the pathogen inside, which means that has a VCR that recognizes the antigen. Why we only want to help this B cell? Because this B cell will now convert into a plasma cell, and the plasma cell doesn't have the VCR, but instead it will secrete a soluble form of the VCR, which is the antibody, okay, which will be exactly the same one that the B cell, precursor B cell had in the surface. So this antibody will bind this antigen, and so will now be able to bind the pathogen and neutralize the pathogen, or fix complement, or opsonize, okay? So it's all connected. Now you're learning like pieces, and the difficult part here is to connect everything. So thank you for the question. Yes?
So just confirm, the T cell only recognizes like a small peptide of
Yeah, correct.
Does that mean the B cell has a mechanism to like degrade the
Oh, yes. And then we have a lecture that is how the antigen is presented in MHC class 2 and MHC class 1. So we'll learn that. Excellent question. I love because you are asking exactly the questions that you should be asking. Yes?
With red blood cells, I know they're not nucleated, so they won't have any MHCs. And I know that a lot of viruses won't attack red blood cells because of the not having a nucleus. But with malaria, because they go into the red blood cells and replicate and die sad.
Yes, malaria.
How would the red blood cells indicate that they're infected?
Excellent. And that's a Plasmodium falciparum, which is the causative agent, infectious agent of malaria, has evolved this evasive mechanism that is able to go and stay as part of the life cycle of the pathogen in the blood cells. And during that part is invisible because they cannot be killed by the CD8 T cells because there's no MHC class 1, and the red blood cells don't express MHC class 2 either, right? So the Plasmodium during this stage is invisible to the T cells. But then the parasite has other stages in the liver where infect other cells, and that is when it can be attacked. But yeah, it's a smart strategy, you know, from the Plasmodium, and that's why, in part, it's a difficult parasite to fight, and we have malaria as a disease. Yes.
But even if once it then infects other cells and it can then be attacked, surely the disease is
good. I don't know the details on how. My understanding is that, but I, take it with a grain of salt because I'm not a specialist in malaria. But my understanding is that there is a stage in the parasite cycle in which it goes into blood cells and then it goes into the liver. It doesn't go, and that's where it starts causing disease, right? And so, yeah, but you may have the parasite may never be eliminated, right? Fully, but yeah. Good, excellent questions. I don't know, Shop, do you know more about malaria and how it gets? I can understand that in the red cells it will be invisible and I will not be eliminated, right? Yeah, absolutely. It's a smart immune evasion strategy. That's a word we use as well. Okay, so do you have the adapter? Josh is not here today, so I was waiting for him, but I need to connect the clicker. So you can go to the flip classroom slides and start with the clicker questions. And so you start on your own, and then we will go very quick when I'm able to connect, okay? Thank you. Okay, guys, you can, I believe that you should be able to start voting now. And remember, it counts now, so don't forget about pressing your clicker, okay? And it's AC. The frequency is AC. Yes? No working. Okay, wait, hold on. I think I know why. I think I know why. I'm sorry. I plugged the wrong thing. Okay, now it should work. So, no? It's not working with AC? Okay. I see 17 responses. So for some of you are working, but I imagine it's not the who for whom is working. Works on the phone, and it should work the other one too. So let me see how I can. Did you try another frequency, AB or AA, and it doesn't work? AC doesn't work? That's very weird. Now it's working? Oh, good. But that's weird. Are you sure AC is working?
Wait, but when you answer, it says no base measure.
Yeah, no, it's not detecting it here. Okay. So I need to. Okay, I will stop it now. In any event, don't worry, I will delete this and I will post it again, okay? So if it didn't work for you, you will have a chance to do it again, okay? So go ahead with the other slides. And so you are, again, we will go very quickly afterwards. It worked last week, so it should be working now. Let's see if it works now. Start. Okay, great. So we will post again this one, and now it's working. Okay. It was just the connection here. I'm sorry. Sorry about that. So why don't you start voting with this one? I'll give you 15 more seconds just for you to press the... Okay. Everyone voted? Okay. Even if you voted before, you need to vote again with the phone, okay? Make sure you voted again, okay? Because I will, this is the start from scratch. Okay, so vote again because I'm going to stop it now. Okay, I'm going to stop it. Everyone voted, right? I just want to make sure there's no misunderstanding. Okay. So one reason that pathogenic microorganisms have an advantage in the host they infect is because they anyway, so this one we did already. I'm putting the wrong. So we should go to the slides. I'm sorry about this, guys. We should go, we can do this one. Have previously been encountered through natural exposure. No, that will not give them an advantage. Reproduce and evolve more rapidly, yes. So the correct answer is B, okay? So we'll move to the next one, and this is true, false, but no, I need to go, wait a second. So let me, we will go to the ones from corresponding to this lecture, which I had them here, but I had to close. Okay. Okay. This one, we need to, we haven't done these ones, right? Okay. Oh, there you go. There's a blank one. Okay, perfect. Sorry about that. I'm very sorry about that. Okay, so let's do this one. And so I give you 30 seconds to vote on this one, since you've been working on this one already. Okay, make sure you voted, and I'm about to stop the clicker right now. Okay, I'm going to stop it now. And so said, what is false about adaptive immunity? Existing in vertebrate and vertebrate organisms, and we learned that adaptive immunity is only in vertebrates, so that's false. Adaptive immunity is orchestrated by specialized immune cells, which are B cells and T cells, which you know that. The right cells are selected from a pool of many lymphocytes to expand and respond. A slow response, but can provide a long-lasting protection. So we'll learn about that. Highly specific response to unique components of the pathogen. The specificities of the adaptive pathogen receptors are acquired through gene rearrangements that we will learn in a couple of lectures during the lifetime of the organism, okay? And so, any question about that? We have talked a lot, so I will move, unless you raise your hand for a question, I will move to the next clicker, okay? So we can cover more. So let's do this one, and I'll give you, let's say 20 seconds for this one, or yeah. And there is this terminology that you haven't learned, but this is false. The A is false, and you will learn about this terminology later. Okay, I'm about to stop this, so you make sure you voted. And so this is also very easy, and we have talked. The process of clonal selection results in the amplification of particular T cells and B cells with a specificity for antigen, correct? Okay, any question about that? We will learn about receptor editing in precise joints. You haven't learned about this yet, but you have learned about clonal selection, so you all answer the right, okay? So let's go to the next one. Okay, so I give you 40 seconds for this one, okay? Okay, I'm about to stop it, So make sure you vote it. Whatever is your answer, doesn't matter. Make sure you vote it. I'm going to stop it now. And so, it's divided. So what I want you to do, and I will post again this right now, I want you to talk to your neighbors and vote again, okay? Let's see to which one it polarizes. Talk to your neighbors and make sure you are in agreement, and if not, convince your neighbor of which one is the right one. Okay, I think it's working. You guys are good teachers. And so let's stop it now. Make sure you vote it, guys. There should be close to 38 here, you know, so make sure you vote it, and I'm going to stop it now. If I can grab it. Okay. Okay, so I'm going to stop it now. It's being complicated. We can start reading, right? So can any of you tell me why D is false? Why D is false? Yes. Why is false? Sorry, yeah. Why is true? So it's false, right? It's false, that's why we select it. But yeah, it's the correct answer. Why is D the correct answer?
So D, because they don't go to the primary lymphoid organs, go to secondary.
Exactly. So that's all. All the rest is good about that answer, okay? And now I try to do it fancy, and I'm having trouble. Okay, so we will read this one, and then I'll post the next poll. So adaptive immunity has evolved systems to recognize billions of different biochemical structures, and this is true. Are the T cells recognizing billions of different biochemical structures? Do the T cells recognize different biochemical structures? Do the T cells recognize lipids? Or right, or carbohydrates, the T cells. What do the MHC molecules present? Peptides. Where are peptides coming from? Proteins. So T cells can also recognize peptides derived from proteins. So now we have another adaptive immune cell. B cells, they recognize antigens in their native form, and so they can recognize many different biochemical structures, okay? And then antigen is any substance, self or non-self, that can be recognized by receptors of the adaptive immune system, and that's correct. You know, the antigen can be self or non-self, whatever is a ligand for the BCR or TCR. Adaptive immunity works through the production of clonally distributed antigen-specific receptors expressed on T lymphocytes and B lymphocytes, okay? That's correct. And then this D, which is the one that is false and should be selected in this question, says a lymphatic system filters the body fluids to facilitate encounter between microbial antigens and the rare, highly specific lymphocytes in secondary lymphoid organs. It should say secondary, because that's the meeting place, the secondary lymphoid organs. The primary lymphoid organs is where the T cells and B cells will develop, okay? Okay, so I think that we can wrap up. Let me do one more, last one, okay? And so I will start the pool. You start doing this one, and with this one we will wrap up the class, okay? So I'll give you, let's say, 40 seconds for this one, and I will start the poll right now. Okay, I'm gonna stop it, so try to make sure you vote it. Please make sure you vote it, and I'm gonna stop it right now. And then we will go through this, the results. All of you answer E, which is the correct answer, and I wanna make sure that it says which one is true about adaptive immune system clonal selection. Each lymphocytes bear a single type of receptor, okay? May have many copies, many hands, but they all have the same shape. Interactions between a foreign molecule and a lymphocyte receptor capable of binding that molecule with high affinity, so it needs to bind with high affinity to a specific molecule, leads to lymphocyte activation, okay? So the BCR and TCR needs to bind with high affinity. Remember this, The BCR and TCR needs to bind with very high affinity to their ligand, to their antigen, for them to be activated. The differentiated effector cell derived from an activated lymphocyte will bear receptors of identical specificity to those of the parental cell from which the lymphocyte was derived, correct? Because it's the same naive T cell that become now the activated effector T cell. Or for the B cell, it's the same B cell that will become the same clone that will become a plasma cell, okay? And then last, it says lymphocytes bearing receptor specific for ubiquitous self molecules are deleted at an early stage in lymphocyte cell development. So they will be deleted in the primary or secondary lymphoid organs? Primary, in development, okay? And are therefore absent from the repertoire of mature lymphocytes. Okay, they are not there. They are not circulating in the periphery. So, and there are more nuance to that, and we will learn that much later. And so, any question about that? Okay, so we will continue with adaptive immunity and more clicker questions on this on Thursday. Okay, take care, guys. Yes. Hi. Let me.
Oh. And I submitted an easy, so you need to approve it for